series. I'm really excited to be here today with you all. And for those of you who are going to be watching remotely or on your own time, I'm really excited to have our speaker today, Dr. Christine Madliger. She's going to be talking to us about From Feathers to Fins. Before we get on to that, I just want to welcome again all of you in way up. I'm talking to you from Windsor, University of Windsor, which is Waitanong, and Anishinaabe Moen. It means where the river bends. And we're talking to you from First Nations people, who are the Odawa, the Potawatomi, and the Ojibwa. Today on our campus, and I've noticed more and more we have a lot of Canada Goose or Mika starting to set up shop, getting ready to teach our students as well as professors. And I wonder how it is like on all your campuses or wherever you're living. It's really excited to talk to you about our seminar series, where when we first started FishCAST, we wanted to have a seminar series, but who are we going to ask? Well, the easy pickings were the principal investigators who were on our part of our FishCAST grant. So they all gave talks, and they were like, okay, well, they've done that. Who else are we going to call in? Well, by then we started to really develop our partnerships in FishCAST. So our other seminar series was made with our interesting partnerships, and they talked to us about their journeys through science and all the different paths that they took. And then after that, we're like, okay, well, we now have our trainees going through our program, and they can now talk to us about what they're doing now and what their journey has been. And so we've switched over to asking our trainees, who are now our alumni, to give talks. And I'm no more excited to present to you again, Dr. Christine Madliger. I've witnessed Christine's journey for a while too, from feathers to fins. I'll have Kristen give you the whole background, but Christine was also really seminal part of FishCAST. Was always supportive at our International Women's Day. She helped co-organize it with our associate director, Dr. Catherine Febria. And so again, Christine was such a seminal part through all of this, behind the scenes as well as in front of the scenes. And we're none so happy when she was able to establish, again, so great, so amazing, her own research program right now at Alumni University. I don't want to take away what Kristen's going to be talking about, but she was so kind to take a moment out of her hectic schedule through teaching and supervision and just being an all-around fantastic person again, to talk to us about her journey as FishCAST actually as a catalyst for research in the aquatic sciences. And I'm really so grateful. I don't want to tear up. I'm like, oh my God, no, there's so much more, Christine, to have built where you are today. FishCAST played a very, very tiny role in the amazingness that is you. So I am going to then allow Kristen to take over, and thank you, Kristen, for moderating this and as well. And at the end of this, we'll have the students just talk to Christine, or trainees talking to Christine, and then I'm going to bow out, so it's a lot more. Let's say, and we won't keep recording, so it's going to be much more intimate, one-on-one. You can ask all the questions that you want to ask. Mm-hmm. And so, but before we do, I just want to acknowledge Izzy's comment in the chat that she had to do a full diversion around the bio building this morning because of the geese. So thank you for confirming that it's not just me they're attacking. So, and with that, okay, please welcome, first of all, Kristen, to welcome Christine.
Yeah, hi everybody. So I met Christine very early on when I started my graduate degree, and we kind of, just as I was starting, she was going on to start her career. But from the short time that we overlapped, it was always all good things that I heard from people in the department, and there was a lot of respect for her. So she's an assistant professor at the biology department at Algoma University in Northern Ontario, and she's received her PhD from the University of Windsor in 2016 under the supervision of Dr. Oliver Love. She then completed an NSERC postdoctoral fellowship with Steve Cook at Carleton University, which she followed up with a FishCAST postdoctoral fellowship with Trevor Pitcher at the University of Windsor, and this was co-supervised by Stephen Cook. Her FishCAST research was completed in partnership with Andrew Drake at DFO, and it focused on assessing transport stress and mitigation techniques for the translocation of the eastern sand darter. But Christine is first and foremost a conservation physiologist, and her research interests include stress physiology, species at risk, aquatic ecology, and determining avenues to make conservation physiology techniques more accessible to a wide community of practitioners. She is also enthusiastic about creating inclusive teaching, research, and mentorship environments for students. And so, yeah, I'm really excited to introduce her today and hear her talk.
Thanks, Kristen. That was really nice. Thanks, everyone, for being here. Thanks for the invitation to be a part of the alumni series. So I'm going to touch on my title first, this, Feathers to Fins. I thought I was making up this very cute title. And as I was preparing to sort of make sure that my presentation had the right vibe, I went on the FishCAST YouTube channel and I went back to some of the previous talks in the alumni series. And there was Brooklyn's talk from a year ago with this portion of her title that was From Fins to Fur. And so after I got over a little bit of the embarrassment that I had personally, and I reflected on it, I thought this was a really great expression of the fact that, FishCAST has welcomed in sort of a diversity of scientific backgrounds in terms of the students that have come through, the trainees that have come through. And it also has given multiple people sort of this opportunity to jump off into research areas that, you might not have had anticipated. And so that's part of what I'm going to talk about today. Okay, so before I get into it, I just wanted to give a land acknowledgement for where I am. So again, I'm in Sault Ste. Marie in Northern Ontario. So I'm on Robinson Huron Treaty Territory, and so this is the traditional lands of the Garden River First Nations, or Ketagonsibee, and the Batchewana First Nations. I always like, when I'm sort of in a room of people that might not be familiar with Algoma University, to just mention that our campus is a little bit unique in that our sort of main building on campus is actually a former residential school. So this was Shingwauk Residential School. So there's a lot of history here. There's a lot of heartache here. And we sort of have this unique connection to our local Indigenous community. And so the land that we're on and that building were provided by Chief Shingwauk and his community. And it has sort of this express purpose to educate people that are in this territory with, a special focus on the needs of Northern Ontario, but also we have a special mission to promote cross-cultural learning as well. And I think, sharing that special mission is just important when I introduce my affiliation. Okay, so the other thing I'm going to do is I'm going to give you a little bit more of a background in terms of like who I am as a person, a little bit of a scientist, positionality statement. I'm a white cis, I'm Canadian and American. I self-identify as a woman. I didn't grow up in Canada. I grew up in, middle-class suburban Ohio, and I was very privileged in that I had access to, green spaces that I could get to just by walking down the street where I live. So I have, a lot of memories of, catching lightning bugs, picking wildflowers, chasing toads, all those kinds of things. And so, from a very young age, I sort of had a heart for and like an interest in nature. But my folks were not very outdoorsy people, right? They liked, the beach and a suntan, but not necessarily a hike. And so it took me until I got into onto an undergraduate program in general science, where I was taking my biology classes in first year to really realize that side of science, like wildlife ecology and nature-based practices and conservation biology, could potentially hold a career space for me. And after my degree, working for a not-for-profit organization. And so this, in hindsight, it really gave me a lot of perspective on the practical aspects of doing conservation work, whether that's time, whether that's the amount of people that you have that can be involved, right? So people power, funding resources, social buy-in, all of those things I got sort of a better perspective on sort of on the ground. And then as I got into my graduate degree, so during my PhD, I started thinking about sort of different conservation paradigms. And there was one that kind of stuck out to me, which is known as new conservation science. So if you're interested in conservation, approaches, You can type this into Google Scholar and you can go down the same rabbit hole that I did. So there's a whole variety of sort of tenets that come along with this approach that are a little bit different than maybe traditional conservation biology as it was, defined in the 80s or so. But the thing that really stuck with me about new conservation is that when we put our conservation science into practice, we have an ethical obligation to not harm people when we do that. And so that's something that struck a chord and that I've tried to carry through a little bit more. And that's part of the reason why over time I've become more and more interested in, really collaborative or more co-created research opportunities and, decision-making processes that consider all the different types of data we have, not just the ones that I sort of was exposed to during my very Eurocentric kind of scientific upbringing. Okay, so my presentation is very much going to be more of a reflection, so less about data. I don't actually have any data in this presentation, and more about the experience. And so throughout it, what I'm hoping to do is offer some little pieces of advice, things that I either benefited from because I learned them along the way, somebody said them to me, or something that came out that I think I would have benefited from if I had maybe heard it earlier in my journey. And so, now that I've given you sort of a little background on me, I'm going to take you back into my PhD. So I worked on this very sweet little bird that's known as yellow research focus of my PhD. I was really interested in whether stress hormone levels, so in birds, corticosterone, whether they could tell us anything when you measured them about sort of the quality of the habitat that these birds were nesting in, the disturbances that can come into their environment or that we can mimic through experimentation, and then whether we could use those stress hormone levels to predict, how well is this bird going to do reproductively in the season? Can we predict its survival from year to year, etc.? And, this bird was a great model organism for a whole bunch of different reasons. But, birds weren't just a model organism to me. I was very much interested in them overall. I volunteered a lot at a bird banding laboratory. I was the type of person that, if I ran into somebody else who wanted to talk about, using feather molt patterns to age a sparrow in the fall, that was an intriguing conversation for me. Okay. But at the heart of what I was doing, my PhD project, really interested in contributing to this field that was still very much developing and still very much is today, which is conservation physiology. And probably everybody in this room is familiar with it. If anybody isn't, it's exactly what it sounds like. So we're trying to use physiological tools, techniques, knowledge to really solve a variety of conservation problems. Because of this interest that I had overall in contributing to the development of this field, what I decided to do was try taking a little leap at the end of my PhD in terms of what I moved into for my postdoctoral work. And so I went to Steve Cook. So Steve is considered sort of a founding scientific figure in this field. He did a lot of work in terms of, defining or codifying the discipline. We had worked a little bit together on some perspective pieces along with my PhD supervisor, Oliver Love, in this space. And And so, approaching Steve, these are the things that I had to offer in terms of experience working in the aquatic sciences. And so this slide is complete. There's nothing missing there. So me, who had only ridden once, I think, in a boat, I had never caught a fish. I had very much kept my feet squarely on land, asked Steve if he'd, essentially be willing to take a chance on me from the sort of experimental on the ground perspective. And I will be entirely honest, at this point in my career, like organismally, I didn't have a great interest in fish, right? If you had laid out, here's mammals, amphibians, even plants, naturally, I would have gravitated to those other things first, right? But I told myself, do it for 2 years. You'll gain some appreciation. You'll get all of the experience working right? And then you can go back to the birds. Okay, and so then how did it go overall? So this was a 2-year postdoc. As Kristen said, I did it under an NSERC fellowship. And so the focus of this work was looking at whether there were physiological and behavioral consequences for fish that get exposed to light pollution. And so I was interested in this. There was a good gap in terms of the information we had at the time, and actually that we still do, in terms of how light pollution is affecting a lot of our aquatic environments and in fishes in general. They've had less attention, even though as human beings, we put our settlements along waterways, and so they're heavily affected, but we have a bit less information on what that means for the species that live there. And so for this project, I went to Queen's University Biological Station. You know, Steve and I planned some great studies on bass and bluegill. So that was sort of carrying over with the stress hormone levels that I had experienced with in my PhD, but then in some new ones that I was excited to learn about, like heart rate. You know, there were some good things that came out of this experience. I absolutely learned new sampling techniques, where I can take blood samples from fish, for example. I got into the behavioral side of things a little bit more, where I really hadn't tread much before. And so I was appreciative to get into that sort of side of the literature and what can be done with those sort of non-invasive metrics. I gained a lot of experience troubleshooting. And then I also had this wonderful opportunity to mentor other students in the lab. And so there's just one that I've pictured here. This is Eli Latcham, who did a nice little study for his undergrad thesis on bluegill responses to artificial light. But then a whole bunch of other things happened at the same time. I had interference issues with my loggers. Failed in both my indoor tank system as well as my outdoor mesocosms that we were using. I lack a natural ability to really catch a fish. And so even though I tried, I was very dependent on other people that were more capable of it to go out with me on the boat and be able to get individuals to go into the studies. And then the other thing is I just had a realization that I'm not really a fish surgeon kind of person. And probably I should have reflected and known that about myself, because I was never good at dissecting anything. So as soon as you're like going inside and rummaging around in the in the animal, it becomes a lot harder for me. And that was for me, like from just like you know, how I feel about it in my body kind of thing, like makes me a little bit queasy. But then there was like ethical things that I started to think about in terms of how well and fast I could do the surgeries in relation to, you know, taking that animal out of the water and having to inconvenience it for that. And so at this point, when you look on paper, I didn't really have a lot to show for myself in terms of progress, and I felt somewhat dejected as a result of that. Now, Steve, being sort of ever a gifted problem solver, thought of Trevor Pitcher, whose experimental hatchery is associated with the University of Windsor, and this is where I was living, so this was right down the street from me. And so he said, why don't we ask Trevor if he'd be willing to collaborate, and then you could work in that space and essentially make up for some of the shortcomings we had in terms of data collection in the field. And so I was very lucky. Trevor welcomed me like fully into this space, physically, professionally, was very kind in that way. And I didn't realize at the time, when sort of this collaboration started, that it would actually be the start of collaborations that we've sort of been maintaining for going on 5 years now. So all of this led to the fact that I had the opportunity to complete some projects that were related to of that progress that I didn't attain previously. And so working in the hatchery was great. I had the ability to combine, you know, some studies in physiology, some in behavior. We could work on lake sturgeon, which was like a little fish that we were looking at, the little juveniles. And so that's a fish, you know, I've fallen in love with in a general sense, as well as red-side days. So the species at risk space was appealing to me. And then so, you know, within the span of that postdoc, I went from feeling like essentially I didn't have a hope to feeling like, maybe I could design and complete and share the results of experiments within this aquatic science space. And so those two little pieces of advice that I think kind of come out of that experience is, you can take a leap. And it really helps when you have supportive colleagues, right? So if you're thinking of changing or pivoting a little bit, when you have a nice group of people that support you, that leap can be worthwhile. The other thing is, don't let a challenging experience, so one experience that you have in the new space, make you feel like you don't actually have the capacity to belong in that space or in an entire field. Okay, so I'm at the end of this first postdoc, and when given the opportunity to keep doing fish work, I said yes. So rather than pivoting back and going into the birds, I decided to keep going in the fish space. And so that's how I got into FishCAST. Steve and Trevor had the capacity as being involved in this program to allow me to apply for a postdoctoral fellowship through the FishCAST program. And so they were my co-supervisors for my project. And then I worked on a different fish for this, which is the Eastern Sand Darter. Eastern Sand Darter are these, relatively small, we're talking about a couple of grams, benthic, freshwater, Ontario population risk. One sort of thing that was happening at at DFO, which was they were planning for a reintroduction program for this fish. And so that's how I got involved in terms of my internship placement or partner as being Andrew Drake, who was one of the leaders in terms of putting this reintroduction program together. And so they were doing a lot of work on all the different sort of moving parts, arms, facets of eventually being able to move these fish from one waterway to the other. And so, this work was taking place in southern Ontario. There were, some good stable populations of this species within the Grand River, but they had been extirpated from big otter creeks, you can see in the map. So the idea was that you could take some fish from the Grand River and you could translocate them and establish some new populations in areas that have, good, sustainable, worthwhile habitat for this species. And to this research program, despite having never met me or heard of me. And so I'm really appreciative of that because it was like this neat opportunity to do some work that could directly inform how a process is going to happen on the ground in terms of conservation approach. Okay, and so what we focused on for sort of my side of this project was the transport aspect of the reintroduction. And so obviously to move these fish from the Grand River into Big Otter Creek, they had to go for a drive. Okay, so somewhere around an hour to 2 hours, depending on how things go in terms of captive time that you would be holding these fish for. And so, something that we knew from the literature and from a lot of work that's been done actually in the aquaculture industry is, transport is a stressor for fish. You see a response in terms of a lot of, quick acting hormonal markers. We see fast things like heart rate and ventilation rate. You can see affects performance or other performance indicators, behaviors, subsequent disease susceptibility. So there are all these things that you're worrying about a little bit when you're thinking about taking these fish from one place and just even driving them for an hour or two to another place. And so then when you add this idea that the fish are going to have to go through this transport event along with going through a capture event, sometimes being in captivity for species that are captively bred, needing to get released into a new waterway that they're unfamiliar with. All of these things can kind of work together and you put the fish in kind of like a multiple stressor sort of scenario. And so ultimately what we want a reintroduction or a translocation program to do is promote persistence of those fish once you release them, and then overall survival so that they can actually establish a population. So you want to minimize the things that act as stressors within that system or that series of events as much as you can. And so sort of a wider perspective in terms of reintroduction of species at risk fishes, this is something, so translocation or reintroductions gets mentioned in a variety of different recovery strategies, but not a lot of fish had seen a lot of movement in terms of actions to put that into place. So this is an infographic that's showing you 14 species at risk fish that have had translocation mentioned as part of their federal recovery strategy. The ones in black have been initiated in some way, and the white ones essentially just have a mention but haven't done anything. Now, I could have filled in that little eastern sand darter could be black now that everything started. But overall, this can be a critical component of sort of implementation of recovery plans over the next decade for a whole variety of different fish. And the thing with the transport event is it represents, like on an hour-to-hour basis. This is, a relatively short time period in the whole process of reintroduction, but it could have a disproportionate effect on overall success. And when you're moving fish in this kind of scenario, as opposed to maybe in an aquaculture or stocking kind of scenario, the stakes can be really high because, you're taking individuals from another population in the wild, or you've spent an exorbitant amount of money captive breeding them. And so making sure that you really pay attention to minimizing the stress, minimizing the potential mortality. This is part of, ethical conservation practice, but also in just making sure that your program is, efficient and successful and worth oftentimes the immense investment that you put into it. Okay, so what we did was we studied transport stress in eastern sand darter. And so we needed to do everything non-invasively. These are tiny little at-risk fish, no blood samples for hormone samples for me. So I took some of that experience working with behavioral metrics and applied it here. And so what we did is we looked at how a simulated transport event influences behaviors that we expected to be ecologically have to be able to avoid predators. They have to be able to find food resources and find new habitat and all of these types of things. So we picked behaviors that we thought would help give us a window into some of that. We also looked at whether if you, after the transport event, allow the fish to have an acclimation period within their production waterway. I don't mean sticking the bags that you transported them in for 30 minutes before you let them go to acclimate them to water temperature. I mean putting them in an enclosed cage essentially for 24 to 48 hours where they're protected from predation. They can recover from the transport event. We looked at whether that could promote behavioral recovery essentially. So this is a process that's known as soft release. Okay, as I said, I'm not going to go into data. I had a master's student take some of this preliminary data that I collected during my FishCAST time, turn it into a master's project. The Coles notes is that the soft release enclosures didn't actually really make any difference. If you want to ask me questions, we can talk to it when we get to the end. However, this experience was very rewarding for me in terms of, contributing knowledge to the development of a program. And so it led Trevor and I to actually collaborate on a whole variety of different projects across different species that were all focused on measuring and minimizing transport stress. So, Ashley Watt and I worked on some of this for red-side dace. Dane Roberts was a master's student that I co-supervised with Trevor, where we did some work in Atlantic salmon. Olivia Galloway was another master's student that we co-supervised, and we did it in the context of lake sturgeon stocking programs. And so, this experience at FishCAST really has now led me to have a major piece of my research program and my lab's objectives as I've become a faculty member at Algoma related back to transport stress, to reintroduction, and to species at risk. And, that FishCAST with the program for about like half a year. It was almost 8 months. So I didn't put up a little banner for advice, but something that was very short had a very defining role for me. So I would say, don't underestimate the potential of, a short project or a short collaboration to provide you with something very valuable that you might take forward for a long time into your career. Okay, another important part that Tina mentioned in terms of my experience in FishCAST was co-teaching the science communication micro-credentials. So I did this with Catherine. And when I think back, so I still take these little screenshots and I put them into my science communication class of some of the projects. The final projects that we received from students absolutely blew me away. Everything from people who produce coloring books to magazine articles, blogs, videos, plans for music. Really sort of inspiring part of something that I got to do during my FishCAST tenure. And the other thing that it did for me was, I never really considered myself, I wouldn't have called myself necessarily like a science communicator or somebody with expertise in science communication. But when I looked back, sort of, when you look on your broader context of sort of all the activities that you've been doing and what's interested you, even back when I was at the not-for-profit, a lot of my time I focused it on outreach, right? Talking to school groups, giving demonstrations, making printable materials that provided information about species that we had on our site. And so I really appreciated that, other people like Tina and like Catherine in the FishCAST program recognized that I had value to offer within this sort of context. And so the success of going through that micro-credential and being able to co-create it with Catherine has formed the basis for now this full graduate science communication course that I teach at Algoma. And then the other thing that I wanted, the sort of last piece that I wanted to reflect on from my FishCAST time is, the people that I worked with during my time there gave me the opportunity to work on some other topics that I was passionate about in relation to, the broader scientific community that don't have anything to do with experimental research. And so the big one for me was, promoting inclusivity within all of our different sort of scientific and professional spaces. Tina and Catherine are leaders in this space, and they really gave me the opportunity to learn from them while I was a part of the FishCAST program. And so, this paper in the bottom left there that we had published, this was a result of a FishCAST workshop at an aquatic conference on the importance of networking and how we can promote equity and inclusion in terms of early career researchers being able to have those opportunities to network. And when I think of ideas within science, one thing that kind of stands out to me is just how collaborative, how long the author lists are. And so this was another time where the people really mattered in terms of who you get to work with and who you can learn from and what you can build. And so that's sort of where this is something that's been like a theme through everything. The people matter. As much, if not sometimes more, in comparison to the exact study system. So that's something that sort of really stood out for me, got really solidified during my time with the FishCAST program. This is often, I think, advice that students receive when they're kind of deciding who to do a graduate degree with. You often get told, you're not just picking the project, you're picking the person or the supervisor. But this sentiment carries well beyond that. So into our professional careers, the people also matter. Okay, and then I'm going to my journey. So this is a picture of a portion of my family. So there's me, my husband, my kids. The other young people are my sister and her husband. And then the two people in the middle are my mom. And then, I don't say stepdad because this person didn't come into my life until I was an adult, but I also hate just saying like, this is my mom's husband, right? Because that also doesn't sum up the relationship. So somewhere in the middle there, but that's Gary. And so for the entirety of the time that I was a part of the FishCAST program, for me, both of these people were terminally ill. And so right at the end of my time at FishCAST, 1 month before I started my faculty job, Gary passed away He had a chronic lung condition. And then 2 months after I started my faculty job, my mom passed away from pancreatic cancer. And so at this, very transitional time in my life in terms of a career, I especially I'm still now today getting out some of the work that I did during FishCAST. But again, for me, it was really the people that made the difference, because I had, encouraging, generous, understanding collaborators. All the difference to me. It's me be able to say that, that dip, I keep calling it that, but like professionally, that happened to me, even defining what my career needs to look like going forward, right? So if you can, if anyone's struggling around you, if you can be like a voice of encouragement for someone, or a problem solver, or someone who invites someone into a new space and makes them feel welcome, that can matter immensely for other people. Okay, and then the last thing, I like to wanted to say was, if you are comfortable, it's good to share the when she kind of reminds other people that we don't often have, very straightforward stories. Some of the things that define our careers are really things that we just stumble upon, rather than something that we necessarily are meticulously trying to build up, right? And that when we sort of value and we build the human relationships, this is often what actually makes the science really enjoyable. And so with that, I'm just going to say thanks to, all of the people that I mentioned and everyone else that supported me during my time at FishCAST and beyond it. It really was, despite being a very short time period, a very sort of important component of who I am as a scientist now. Thanks for listening, everybody.
That was amazing, Christine. Thank you so much.
Oh, thanks. You're welcome.
Thank
I'll
you.
stop sharing.
And thank you for sharing all, like, your entire journey, the good and the bad. And the reason why you are where you are today, Christine, is that you had support and collaboration, but you also had the resilience and the spirit, and that and the personality type that you are. That speaks for itself as well. I don't want to, like, you're so kind to always put others, but please take that onto yourself, that it's your strength and resilience as a person, as well as your capabilities, your expertise, your kind-heartedness, your generosity. And I want to say that this is an amazing theme for International Women's Day this year, which is give to gain. And I think you really embody that, Christine. And so I wanted to thank you again. And so the theme is to uphold others through whatever kind of means, is through the collaborative spirit. And I see you're so always so generous and acknowledging others, but you've done that for others as well, Christine. And so I want to also recognize and acknowledge that as well. You're an incredible person and component of others people's progress. And so this give to gain is very much a reciprocity that I see happening. And so I just wanted to just put that out there and say thank you with deep gratitude. Nice. Yeah, of course. Okay. Anyone have any comments or questions for Christine before I log out? But I might want